A workflow for fluxomics.
As an application of the so-called stable isotope-resolved metabolomics, the objective of our workflow is the estimation of reaction fluxes, by fitting model predictions and experimental mass spectrometry measurements of 13-carbon propagation.
A quick introduction about mass spectrometry and fluxes.
In the context of 13-carbon labeling, isotopologues are mass isotopomers. This is isotopic isomers of different masses depending on the different number of 13-carbon substitutions. In the example, labeled glucose propagates through glycolysis to lactate. Lactate, with 3 carbons, has 8 possible isotopomers. The 8 isotopomers can be grouped in 4 possible isotopologues according to the mass. Isotopomers with 0 labeled carbons. Isotopomers with 1 labeled carbon. Isotopomers with 2 labeled carbons. And finally for lactate, isotopomers with the 3 labeled carbons. However, in our simple example, just can be produced 75% of isotopologues with 0 labeled carbons and 25% of isotopologues with 2 labeled carbons. This is a restriction imposed by the reaction stoichiometries and the specificity in the transitions of carbon positions from substrates to products. However, the situation becomes more complex if an additional pathway is added, in the example the oxidative branch of pentose phosphate pathway, where one of the labeled carbons is lost, then resulting in the appearance of isotopologues with 1 labeled carbon. Interestingly, the abundance of isotopologues with 1 labeled carbon will depend on the relative fluxes through glycolysis and pentose phosphate pathways, then allowing indirectly for the estimation of these fluxes, by comparing model predicted and experimental isotopologue abundances. For example, an increase in the flux through the oxidative branch of the pentose phosphate pathway will result in an increase in isotopologues with 1 labeled carbon. Then, indirectly, by measuring this increase, we can hypothesize about an increase in the flux.
An example.
As an example, is Houston study from the metabolic database Metabolites. In this study HUVEC cells were incubated for 40 hours in the presence of the label in glucose. Glucose, which propagated to glycogen, RNA ribose, and lactate. In this example, the inappreciable abundance of isotopologues with 1 labeled carbon for lactate anticipates a very low flux for pentoses. Following an independent methodology, based on the measure of initial and final concentrations of glucose and lactate, we have estimated a value around 0.45 glucoses consumed per lactate produced, which is the expected for a cell under hypoxia. Considering that glycolysis and pentose phosphate pathways are accompanied by alternative inputs, glycogen utilization, and an extra input of pyruvate, and taking into account that all fluxes have unconstrained values, applying mass spectrometry, a similar estimation will be obtained. The answer will be provided by running our workflow for fluxomics. For the development of workflows for fluxomics based on Galaxy. First, separate tools have been developed. RAMID, MIDCAR, ISO2Flux, and ISODIN, covering complementary parts of the study of metabolic networks based on 13-carbon mass spectrometry. Second, containers have been created for these tools, and further, created wrappers for them to be available in Galaxy. These tools, together with the ProteoWizard container, have been used for the creation of 2 workflows in Galaxy. Of the 2 workflows created, one has been created for steady-state fluxomics, and the other for dynamic fluxomics. This tutorial is focused on the workflow for steady-state fluxomics. The workflow for dynamic fluxomics will be available very soon.
An important remark about standards.
Stable isotope-resolved metabolomics experiments are labor-intensive and sensitive to experimental deviations. Care must be taken recording experimental conditions and results according to regularized standard formats, so, comparison between experiments may be considered. Standards are essential tools in the process of information, communication, publication, and preservation. They ensure data are findable, accessible, identifiable, and reusable. In short, it is about making data fair. To accommodate the needs of these studies, the ISA format is used to report study setup, while raw data are standardized to MZML and NMRML formats for MS and NMR respectively. A dedicated data matrix extension is now available to specifically structure mass isotopolog and mass isotopomer reports, and is integrated to RAMID, MIDCAR, ISODIN and ISO2FLUX tools.
So, implementing standards does not have to be taxing.
Simply use phenomenal workflows, like this for fluxomics. This scheme provides a detailed description of the workflow. RAMID is a tool for reading raw mass spectra contained in CDF files, which follow in standard format for raw 13-carbon mass spectrometry measurements. Raw data are converted in signal intensities. MIDCAR is another tool, based on R, for data correction for natural isotope enrichment. Uncorrected signal intensities are transformed in normalized isotopolog abundances. Finally, corrected isotopolog abundances are the input used for ISO2FLUX, a Python-based tool. This program estimates reaction fluxes by fitting model predictions and experimental mass spectrometry measurements. An additional tool will be added in the future, which will allow for directly reading mass spectrometry data from databases, in particular the metabolites database. Regarding data, two groups of data are required as inputs. Mass spectrometry data, including CDF files, grouped and compressed in a zip file, and an associated matrix of data correspondences, where are recorded the applications of RAMID and MIDCAR, and it is an input of ISO2FLUX. And a complete model description for ISO2FLUX, including both a description of all involved reaction stoichiometries, and the transitions in carbon positions. Also, an optional file can be added with analysis options. Finally, two groups of data are returned by ISO2FLUX as outputs, including the best fit and estimated fluxes. Of course, for the analyses of comparable cases in data collections, at the beginning an iterative loop must be established with the user at least for one representative case, which will modify the model description and other analysis options until a satisfactory fitting is accomplished. Once the correct model and analysis options are established, then the automated analysis can be applied for the rest of cases. For mass spectrometry data, a file is provided with the associated matrix that contains correspondences between istotopolog abundances and signal intensities, and with CDF files, targeter with additional descriptors. This file is a subset of selected columns from the original mass isotopomer distribution matrix, that follows the dedicated data matrix extension available to structure mass isotopolog and mass isotopomer reports. The convention standards applied in this mass isotopomer matrix are currently under refinement. The model description is provided by three files, a SBML description of the model, with all metabolites involved, and the reaction steps included with their corresponding stoichiometry. An additional file, with the propagation rules accounting for the transitions in carbon positions between substrates and products, and, including also, the correspondence between model metabolites and the measured isotopologs. And finally, a file accounting for reaction fluxes, with fixed upper and lower bounds. Usually, upper and lower bounds for external exchange fluxes are fixed. In our example, a glucose uptake of 1.5 picomoles per cell per hour was measured. When two or more are measured, This helps to additionally constrain the relative values of unknown reaction fluxes. In our example, we have fixed the value of lactate production to 1. This will allow us to refer the rest of the values to the production of lactate.
Running Galaxy.
A demo video can be downloaded with the proposed example analyzed, with a detailed explanation of all required steps.
The outputs.
The analysis of the output files will provide the following information. The outputs include, on the one hand, a file with the reaction fluxes that provide the best fit. Together with other file with the isotopolog abundances, predicted using the best fit fluxes, and compared with those measured experimentally. Chi-squares are provided to statistically assess the quality of the fitting. On the other hand, confidence intervals are provided, around each estimated reaction flux, which gives a measure of the significance of the fitted fluxes. These data are provided in the form of a SBML version of the model, with the confidence intervals included as upper and lower bounds, for values of the reaction fluxes. The equivalent information is shown here as a table, for simplicity. An analysis of the results shows that most of fluxes for internal reactions are well estimated. As anticipated, a low value is estimated for the oxidative branch of the pentose phosphate pathway. And, regarding the estimation of the uptake of glucose, a value ranging from 0.43 to 0.45 is obtained, which is close to the value of glucose consumed per lactate produced estimated by an independent method.
The flow for fluxomics using Galaxy, and this tutorial, have been developed in the framework of the project Phenomenal, funded by the European Union.